Okay, welcome back. We ended the last video talking about actin, which is our thin filament. We ran a little bit out of time, so just to reiterate, it has two regular proteins associated with it, tropomyosin, which is arranged in a double strand as well. So this is this orange strand right here, as well as troponin. It has three molecular subunits. So here, 1, 2, 3, 1, 2, 3. It's called the troponin complex. This is our thin unit. The troponin has actin, tropomyosin, and calcium associated with it. So it will bind to either actin, tropomyosin, or calcium. And each strand of tropomyosin covers about 7 G-actin subunits of the F-actin strand. So it follows the double helix and it covers about 7 of these. Okay. Let's talk about if we're in a relaxed state. If we're in a relaxed state and skeletal muscle is calm and relaxed, there's not enough calcium in the cytosol to bind to the third molecular subunit of troponin. We need calcium to bind to that third molecular subunit in order for the steps of muscular contractions to play out. So we must increase the intracellular calcium content. This will actually cause the tropomyosin strand to shift, exposing the myosin binding sites on the actin. Now the myosin can then bind to the actin. So let's go over that really quick. Here, in this troponin complex, we need calcium, an increase in calcium in the cell, in order to bind to one of these troponin units. The third molecular subunit of troponin, we need calcium to bind to. In order for when that happens, that will cause a shift in the tropomyosin strand. So it will actually twist and shift, exposing the myosin active site on these actin molecules. So ultimately, we want to shift the strand out of the way, so then the myosin can grab on to those actin subunits. And when we grab onto the actin, that's what's going to cause that power stroke and actually cause muscular contraction. So we want that binding to occur. So calcium's a huge player in order for that to occur. If we don't have enough calcium in order to bind to the molecular subunit of troponin, it's not going to shift this tropomyosin strand out of the way, and then myosin cannot bind to the actin. And then therefore we cannot have muscular contraction. Okay, so now we're going to shift from the thin filament to the thick filament, and this is our myosin strands. We have a light meromycium, which are rod-like tails, so we have, which include the myosin head, and we have the heavy meromycium. This gives us our cross bridges, right? So we have our heavy meromycium, and then we have our light meromycium, which are our rod-like tails. The globular heads, these globular heads actually are the ones that attach onto the actin active site. If we can get the myosin to bind to the actin, then we can create a power stroke. And a power stroke is what's going to be ultimately our muscular contraction. So here, we want those globular heads to attach to the actin here, once those active sites are exposed. That's what we'll attach here. All right, so let's talk about our power stroke steps. A power stroke can be divided into 4 independent steps. 1, attachment, which is binding. 2, the power stroke, which is bending, which is actually the pull. The detachment, which is the breaking. So breaking apart, so we're going to release. So actin and myosin are going to release. And then 4, the start of another cycle, bouncing back up. So ultimately, if you've learned in undergrad or in any other exercise physiology class, you may have heard the terms binding, bending, breaking, bouncing, and that is ultimately our muscular contraction steps. So we have the attachment phase, where myosin attaches to the actin, the power stroke, where the myosin pulls on the actin strand, and then we have detachment, where the myosin and actin detach from one another, and then boom, we bounce back up and start yet again another cycle. Okay, so now we're going to talk about this binding, bending, breaking, bouncing steps right here. The myosin heads attach to the actin by breaking down ATP to ADP and a phosphate via myosin ATPase. That myosin head formed a cross bridge, right? This is why it's called cross bridge cycling. that myosin head forms a cross bridge on the active site of the actin filament. The cross bridge pulls the actin, which slides over the myosin, known as the power stroke. The release of ADP completes the cross bridge movement, and ATP attaches to the myosin, breaking the actin-myosin cross bridge. Every time ATP is split into an ADP and a phosphate, the myosin head cocks back into place to form another cross bridge with the actin. So this is where we have continuous muscular contraction steps. We need to bind, break, bounce, and binding, bending, breaking, bouncing. We need to do all of that multiple times to have continuous muscular contractions. So up here, we can see the steps. So myosin, we're going to hydrolyze ATP, and we're going to break it down into an ADP and a phosphate. This is what allows us to attach. Requires energy to attach actin by breaking down ATP and a phosphate via myosin ATPase. This is where the myosin heads attach to the actin. So now we're down here, and we can attach to those actin molecules. Now we're going to go down here, and we're going to pull the engage in cross-bridge cycling, which the cross-bridge pulls the actin, which slides over the myosin. So we're going to pull it here, known as this power stroke. The release of ADP completes the cross-bridge movement, and ATP causes it to detach. So right here, we need ATP again in order for myosin to detach from the actin up here. And then we're going to be re-energized to cock back into place and ultimately get ready again for another muscular contraction. Okay, so here is a diagram from start to finish. We're going to go over the steps from the action potential through muscular contraction. So an action potential in a motor neuron causes acetylcholine to release in the synaptic cleft, which we went over all those steps. So acetylcholine released in the synaptic cleft. Acetylcholine is going to bind to those alpha receptors on the cell membrane, on the muscle fiber, on this postsynaptic side. So we're looking for those alpha receptors, which the acetylcholine will then bind. That signal, once we generate an end plate potential, that signal travels down, right? It's going to travel down the cell and activate calcium channels. Calcium is released from the terminal cisternae of the sarcoplasmic reticulum into the muscle fiber, right? So we have our sarcoplasmic reticulum, right? So the signal is going to come down the cell, or action potential is going to come down the cell, and that signal is going to increase the release of calcium from the terminal cisternae of the sarcoplasmic reticulum. Now we have all this excess calcium now in the muscle fiber, which is good because that's what we need, right, in order to shift this tropomyosin out of the way. So calcium then is going to go down and bind to the third molecular subunit of troponin down here, and it's going to cause those tropomyosin strands, which are those double helix strands that follow the helix, cause those tropomyosin strands to shift, now exposing the myosin active sites on the actin, right? So now we're allowed to bind with myosin to actin. We can bind. So that's a key player again. We increase the calcium from the release. That action potential releases the calcium from sarcoplasmic reticulum. That increase in calcium then attaches to the third molecular subunit of troponin. That allows to provide that conformational shift of the tropomyosin strands out of the way, exposing the myosin binding sites on the actin strands in order for myosin to bind to the actin. Myosin heads attach to the actin by breaking down ATP again to ADP and a phosphate via the enzyme myosin ATPase. The myosin head forms a cross bridge on the active site. Right here. Here we have the cross bridge, known as the power stroke, which will pull it across. The release of ADP completes the cross bridge movement, and ATP again attaches to the myosin, breaking the myosin cross bridge. So we need to break this apart and get ready again for the next muscular contraction. Every time that ATP is split, into an ADP and a phosphate. The myosin head again cocks back into place and to form another cross bridge with the actin. Now, this has to occur every single time you want to contract a muscle. And just think, sitting there, think how fast you can move your finger or a toe or any sort of body part as fast as you can. So just think how fast everything has to go back into place and get ready for that next contraction. We have to have all of these series of events coming down the axon, this acetylcholine activity, all of this coming down, signal propagating down into the sarcoplasmic reticulum, releasing that calcium so it can bind to the troponin so we can shift these strands out of the way so then myosin can attach and all of that good stuff. This has to happen every single time you want to have muscular contraction. And what you don't realize is this is continuously doing that to sit you upright in your chair or so you can stand up or so forth for your postural muscles. But it's pretty crazy how fast, so you can kind of appreciate how fast this all has to occur within our human body in order for us to keep exercising or just muscular contract in general. So when we release all of that calcium and that attaches to the third molecular subunit of troponin and everything shifts out of the way, everything's got to go back into place, like I just said. So we have different pump systems and so forth. We're not going to get into the physiology of all these pump systems as well as the cleansing of these alpha receptors. There's another whole set of thing we could talk about cleansing these receptors and all the physiological steps that have to do that in order for everything to be put back in place so we can respond to the next stimulus. For the most part, as much as this seems very detailed, this is still a very brief summary of actually what technically happens at the motor end plate. So we have all these pumps that we need to pump calcium back in order for the signal to come back down so we can release calcium again and do this all over again. So hopefully you can get an appreciation of just how fast our bodies actually work with regards to ions and neuromuscular junctions and so forth. I think it's actually pretty crazy. So all of these things have to happen and go in reverse before we get ready for the next one. And don't forget, this is just the axon terminal and the acetylcholine portion, then all the way down through muscular contraction. But don't forget, in order to generate that end plate potential as well as propagate an action potential, we have to do that whole first step of depolarization, of where we increase the positivity inside the cell and we alter the membrane permeability to actually propagate that signal. And then on that side of things, everything has to go back in place too. And we have all these pump systems working in order for us to get ready for the next signal so we can muscular contract again. So you think you can contract extremely fast, but then when you actually stop and think about it, how fast your ions and all these things are moving in order for you to do that is actually pretty crazy. Okay, so our next video, we're going to be talking about muscular force, recruitment, summation, as well as what a tetanic contraction is. The last video is just finishing up our last few slides. Okay, so stay tuned and go to our last video of the lecture.